Hey guys, so at this point you're probably entering into the lab segment of the Intro to Mendel assignment. So you can see that here on your Unit 5 Division in Genetics. We're going to go ahead and explore that. This video is going to help you to figure out how to use the simulation to go ahead and collect the data that you need in order to respond to the that simulation portion of this lesson. So the first thing you're going to do is you're going to go down to your syllabus and general class info, and you're going to access the Using Clever to explore learning. We're going to go ahead and use the Clever portal to get back to our gizmo so that we can use the gizmo to do Mendelian genetics. Now, I normally don't like to do so many gizmos all at once, but gizmos are a great way to show Mendelian genetics at this stage, and it's difficult for us to all grow pea plants physically in a classroom when we don't physically have one. So let's go ahead and click on this link to Warwick. You'll go over to Students, go down to Clever, click on Clever. It should bring you up to the Clever login. Use login with Google. I, of course, will use my account. You'll use yours. And once it logs you in, you should see a page that looks something like this. And we're going to go ahead and scroll down to Gizmos Pilot. Click on Gizmos Pilot. And of course, for you guys, it'll take you directly to your classroom, or you may have to type in my name or indicate which class you want to go to. You should go to AP Bio 5, and you'll find Mouse Genetics 1 Trait. Go ahead and click on that, and you can start the gizmo by clicking on Launch Gizmo. So once you're here under Launch Gizmo, what you're going to do is you're going to start breeding mice. Now, if you go to Mendel and Mice, which is what you're probably looking at here, you'll read through the introduction, talks about Mendel, sort of sums up some of the stuff said in the lesson, and then you have the actual exploration, the mouse genetics. So it says here in number 1, and I'm not going to go through the whole thing, but just sort of give you guys an idea of how to do this. Drag 2 black mice to the parent 1 and parent 2 boxes. So the parent boxes are right up here at the very top. You're going to drag your mice up there to the parent box, and then in order to actually make them create offspring, you would press breed. Okay. So down here, it might ask you to do that. It'll ask you to describe what they look like. Click breed several times. What's the phenotype of the offspring now? So we can come up and press breed a couple of different times. They're all still black, of course. Do you think the mouse offspring will always look like their parents, etc., etc.? Okay, so you're going to do that. Now, if they're, and then it says press clear, and you're going to drag a black mouse and a white mouse into the parent thing. So we press clear and do this. Now, the next thing it's going to ask us is, of course, to breed these two guys, and when we breed them, we end up seeing offspring that all favor one of the mice. This will give you a clue as to which one is dominant, of course, because the dominant phenotype is always expressed when one copy of the dominant gene or allele is around, right? And so hopefully you understand that based off the lecture, and you can guess which color is dominant. Now, if you're not sure, however, if you want to know more about the genotypes of the mice, you can press Show Genotype, and you can go down and hover over a mouse, and it'll say that for fur, each one of these has one copy of the dominant allele, or capital F, and one copy of the recessive allele, or lowercase f. If we look over here, the white mouse up here only has lowercase f's, and the homozygous mouse over here for black has both capital F's. Now, if you wanted to breed any two of these mice together, let's just say you want to do that, what you would do is you go ahead and grab these mice and put them into the holding cages, and then go ahead and press clear. Once you press clear, you could then take two of these heterozygote or hybrid mice and put them into the parents, and you go ahead and breed them together, and it would show you the ratio of offspring that would result from this. Now you can breed them as many times as you'd like and go through and press that. Now, show statistics. What's cool about this is as I'm breeding them, it's going to be keeping track of how many of each type of mouse that I have. And then we'll show an approximate percentage of how many mice have which phenotype. So for instance, if I keep breeding them, let's say I want to breed up to 500 mice or something like that. I'll breed so I've totally bred 500 mice, and approximately 78% of them have been black and 22% of them have been white. So that can help me to figure out, again, whether or not that's going to be a good indication of Mendel's golden ratio. Now, one thing I do want to make clear is that the ratios that you get here, just like in real life, are not going to completely match Mendel's ratio. 3 to 1 is just a ratio that Mendel came up with after many, many trials. And it's close to what we're seeing here, but of course these numbers are not always going to be exact. You can think of it this way. When you flip a coin, you have a 50-50 chance of getting heads or tails, right? Now, that doesn't mean that if you flip a coin 10 times, 5 times is going to come up as heads and 5 times is going to come up as tails. Sometimes you might get all heads or all tails. But it does mean that if you flip them enough times, so say you flip that same coin like thousands of times, eventually the number of heads and tails that would come up during that flip, assuming it was a balanced coin, would be about 50-50 heads to tails. So that's really all Mendel's ratios mean. They're not exact numbers, but they are predictions in useful percentages to use to sort of guide your data as you're looking at stuff in real life. But of course, recognize that it won't always match exactly to those numbers. Okay, so you're just going to go through that and you're going to answer these questions as you go through. The questions are fairly non-invasive. Hopefully they will make a lot of sense. Just follow the instructions and go ahead and reflect at the very end. Hopefully this simulation will give you guys a better idea of Mendel's experiment and how genotype and phenotype ratios work. Again, genotype literally just refers to these letters that you see here. For fur, so big F, little f would be this mouse's genotype, and its phenotype would be that it is black in color. Phenotype is what you look like based off of your genotype, which is your letters or alleles that you have received. I hope this video has helped you guys and that you enjoy this simulation. We will not be doing a simulation for the next one, so don't worry. And I hope you guys, this helps you to understand Mendel. All right, guys, have an awesome day.